Hello students, this is Dr. Ashish Dwivedi again with you, and in the last lecture I have discussed about movement of proteins into the endoplasmic reticulum and other vascular organisms. Uh, just for a reminder, just for a recall, we have discussed movement of the protein in the rough endoplasmic reticulum, mitochondria, chloroplast, nucleus, and in the cytosol as a short review. So, starting from the last lecture, we have discussed about the targeting sequences, which are 6 to 12 hydrophobic amino acids, and then receptors, and then translocation channels or the translocons. Right now, I'm starting a detail of the transport of proteins into the ER lumen. So, to understand this process, how protein moves into the ER, we should look at the cycle which is known as signal recognition particle cycle. This signal recognition particle cycle is given on page number 589 in the 8th edition of Lodish. As in the last lecture I have told you that I am taking a reference of this book because this book, I love this book in a very simple language. It has a very good language. It is written, well written and well composed. So here you can see in this figure the 5 mRNA. On the mRNA, ribosomes are there which are translating mRNA N terminus to C terminus, and near to the N terminus there are the signal sequences, and these ribosomes have a SRP binding site. SRP binding site. This is SRP, signal recognition particle. What is this signal recognition particle? In very short question, or to consider it as a question, it is a ribonucleoprotein complex made up of 300 nucleotides, 7 sLRNA, and 6 different proteins. So to get clarification for this fact, you should consider the figure caption of Leringer, which is given on 1141 in chapter number 27, protein targeting and degradation. It is well written in the caption that the SRP is a rod-shaped complex, contains 300 nucleotide RNA, 7 sLRNA, and 6 different proteins. Molecular weight 3,25,000. One question. Another question. It's one protein subunit binds directly to the signal sequences, and its function is inhibiting elongation by steric blocking the entry of amino acyl tRNA and inhibiting peptidal transferase, which is a part of the larger subunit of ribosome, larger subunit of the ribosome. So these two questions: what is a structural configuration of SRP? What is the function of the proteinaceous part of the SRP? And another protein subunit of the SRP binds and hydrolyzes GTP, means it has a GTPase activity. And the SRP receptors, in the last lecture I have already told you that it is a heterodimer made up of alpha and beta subunits, both of which binds and hydrolyze multiple GTP molecules during this process. That means this process is highly energy driven, requires GTP. So continuing with the Lodish, we can see that the proteins are directed to the SRP receptors where its signal sequences binds. So you can have a look over here that 2 GTP are binding. One is on SRP, another one is on alpha subunit of the receptors. Both are hydrolyzed, but unless they are not hydrolyzed, the translocons are in the closed configuration. Once these GTP are hydrolyzed, translocons are open, protein moves inside the ER lumen, and membrane bound signal peptidase membrane bound signal peptidase cleaves signal sequence. Remind my last lecture, I've told you with the reference from the Leringer, the cleavage point is always on the C terminus of glycine and alanine. So once this protein is cleaved from its signal sequences, it moves inside and get folded. So it is very much clear that the protein moves inside by the cleavage of the signal sequences, and now it get folded. In relation with that, we can also consider section C type questions of CSIR. So have a look on the section C type questions in the Lardish. That's what I love this book, because it has a very good illustration of the experiments. So there is one experiment which is given on page number 587 on the 8th edition. Mind my words, mind my things. It's the 8th edition molecular biology. So on this page, you can see there are the two experiments are given in very short and summarizing language. You can see there is a mRNA on ribosome on the ER lumen, but essentially the proteins are labeled. Proteins are labeled. How the proteins are labeled? Obviously with the amino acids. And amino acids, what are the amino acids? Most frequent amino acid. The frequency of the amino acid which has the highest frequency to be found in any protein is leucine by the 9%. You recall Leringer chapter number, that chapter is amino acid and protein, and you can have a look on the table on which PI, PK1, PK2 and frequency of percentage frequency of the amino acid is given. So on this table you can find what is the frequency of the leucine. So you can also radiolabel cysteine, you can also radiolabel methionine, but it is very good to radiolabel leucine. Once this protein is radiolabeled, now this ER is homogenized. Why this is homogenized? Because it is very tough to isolate intact ER. It is well written in the text also. You can have a look on page where it is given that to delineate the step of the translocation process, it was necessary to isolate ER from the rest of the cell. The isolation of the intact ER, which is delicate lace-like structure, and its interconnectedness with the organelle is not feasible. So what we do? Scientists have discovered when the cells are homogenized, rough endoplasmic reticulum break into the small closed vesicle with the ribosome on outside, termed as microsome, and it requires very high RPM centrifugation. You can study Wilson and Walker principle of biochemistry, and in the centrifugation chapter a clear-cut diagram is given on which RPM microsomes or the ER can be homogenized and to isolate the microsomes. So once we consider the microsomes are the vesicular system with a smaller quantity, with a smaller size of the ER. More or less the microsome properties or the ER properties are the same. So here we can find that the ribosomes are transferring radiolabeled protein into the microsome vesicle, and here you can go with the two kind of steps. One, in the step 1, in a pathway 1, you treat these microsome with the detergent. Once you treat microsome with the detergent, it perforates the membrane of the microsomes, and after that addition of the protease percolates inside the microsome vesicle and digests the secretory protein. So the radioactivity is lost, because the amount of radioactivity you will get in a linear chain, not in the fragments of the secretory protein, because it gets dissolved in the system. But if you don't add the detergents, protease cannot percolate within the membrane, and you can easily get the radioactivity within the vesicle. So what you can conclude by this experiment? Simply, three questions you can fetch. Number one, homogenization of the ER at the very high RPM. You can isolate microsomes, which are smaller form of the ER with the same property, more or less same property. And secretory vesicles, secretory proteins are always found in the ER, not in the cytosol. So we can conclude from this experiment, translation and translocation of the ER resident proteins is tightly coupled. How? In another experiment it is also discussed again. So in the cell-free protein synthesis, when there is no microsome is present, your protein is synthesized along with the N-terminal signal sequences. Once the complete protein is synthesized with the signal sequences, you added the microsomal membrane, but your protein doesn't enter into the vesicles. That means you simultaneously need microsome to have secretory protein within the microsome or the ER lumen. In the second set of experiment, what we have done, what the scientists have done, the cell-free system, protein system was done in presence of the microsomes. So as the protein is synthesized, it moves into the secretory vesicle and its signal sequences are cleaved. So here it is clearly written, there is a co-translation transport process. Means starting protein translation from the ribosome, it need ER lumen to transfer immediately into the vesicles. Otherwise it will not be transferred. So here it is written over here. Here it is written, the protein synthesis carried out in the cell-free system contains a functional ribosome, and to which the secretory synthesized in absence of the microsome, and this is translation and translocation occurs simultaneously. This is a clear-cut question which can be asked in CSIR exam. You need to focus it. All the students should note it down. All secretory processes requires a luminar vesicle to get transported immediately, otherwise it will not be transported. So once this protein is transported within the vesicle, the modification which is known as glycosylation takes place. During the glycosylation, what happens? In the glycosylation, several kind of residues are added, several glucose residues, mannose residue, N-acetyl glucosamine residues are added over the protein, and especially on the asparagine residue near to the N-terminus. So I'm just skipping few of the content because I thought this glycosylation part is quite important for you people, and I can discuss further another things, how it happens and happens. So in this content, when the glycosylation is taking place, there are two type of carriers are used. One is UDP carrier is used, another one is GDP carrier is used. UDP carries N-acetyl glucosamine. Two N-acetyl glucosamines are transferred over the dolicol phosphate, which is an isoprenyl derivative, and UDP transfers glucose. UDP transfers glucose. GDP only transfers mannose, and this process takes place in two steps, one in the cytosol, another one in the ER lumen. In the cytosol, two N-acetyl glucosamine residues are transferred and 5 mannose residues are transferred on outside of the cytosol. Here you can find one question. This first residue, that is N-acetyl glucosamine transfer, is blocked by antibiotic tunicamycin. This tunicamycin blocks transfer of the N-acetyl glucosamine onto the dolicol phosphate. So If you look at the structure of the tunicamycin at the page number 1141, you will find a very interesting structure. This tunicamycin also contains N-acetyl glucosamine and uracil with modified 5' end with the tunicamine, and this tunicamine is also modified, this residue is modified by the fatty acid side chain. So with this structure you can draw a conclusion that once the tunicamycin is available, it do not allows the entry of UDP bound N-acetyl glucosamine, and it is antibacterial agent, so it is very easy to understand. It blocks the protein glycosylation, and in the later sections you will get to know that once the protein is not glycosylated, it is not functional. So protein is glycosylated in the cytosol as well as in the ER lumen. Finally, a complete precursor of the protein, which contains 3 glucose residue, 9 mannose residues and 2 N-acetyl glucosamine residues, which is found in the ER lumen. So this ER lumen contains glycosylated proteins. So another look, you can have a look on another page. In the larynga, you see that this glycosylation is taking place on the asparagine, which is called as N-linked glycosylation. This N-linked glycosylation is the first step of the protein glycosylation in the ER lumen and mediated by dolicol phosphate. One very good question in the recent times has been raised, especially in the JNU examination or so on, that is how the glycosylation of the protein can be identified. So if you see a book, Wilson and Walker, in which page number 336, the protein structure, purification, characterization and functional analysis, you will find answer of this question. It is well written over here that if a protein is glycosylated or it is a glycoprotein, it can be stained by cationic dyes such as Alcian blue. Note it down. Glycoproteins can be stained by cationic dyes such as Alcian blue because it binds to the negatively charged glycosamine glycan side chain or per iodic acid Schiff reagent that is known as PAS. Okay, so this is very clear. On the other hand, there are 3 other steps. If somebody wants to identify the type and amount of monosaccharide, so it can be achieved by hydrolysis of the protein in the methanolic HCl at 80 degrees centigrade. You can easily isolate monosaccharide, and that this reagent will release monosaccharide, and which can be identified or quantified by the gas chromatography. Second point is very important, oligosaccharide profiling. Oligosaccharide side chains are released from the polypeptide backbone either chemically, for example, treating proteins with the hydrogen, especially glycoprotein with the hydrogen, releases N-linked oligosaccharide, or you can treat it enzymatically with the PNGase F, which cleaves sugar on the asparagine link. So this is a case how you can separate N-linked glycosylation within the protein. And for the O-linked glycosylation, which takes place on the serine residues, you can treat the glycoprotein with the O-glycanase, which cleaves O-linked glycans. So in this very short video, I must say, I have tried to cover few things. Let me revise at once. One, how protein is translocated into the ER lumen, and it can be proved by an experiment, by two experiments, better to say. One experiment in which in one experiment we have homogenized ER membrane into the microsomes, and then it was treated as a vesicles. So these two experiments and SRP cycle, function of the SRP, structure of the SRP, how it functions, one unit, that is P54, how it functions. So these are some sort of questions you need to take care of. So I recommend, these are the one kind of retailed questions one should consider for CSIR. Before finishing my this lecture, this assistance to you, I recommend read books. Without books, there is no research is possible, because ultimately in the lab you have to read the research papers. So once you start reading research papers, you will realize your standard of your knowledge. So thanks for today. See you tomorrow with the same frequency. Bye bye. Good day.